Hello, yeah. Yes. Right. Okay. Well, you can tell OCR that it's not right and I don't like it. Right. Make sure it happens. Sorry, guys. Right, so in this video, we will be talking about translocation in plants. Now, it's part of the wider topic on transport. We have talked about the transport of oxygen and carbon dioxide in the blood in animals. But what we're going to talk about today is the movement of sucrose in plants, okay, and how that happens and why it is important that it does happen in the way that it does. All right, guys? So, just a quick summary, a background on sucrose. Sucrose is actually a product. It's a disaccharide of glucose and fructose joined by a condensation reaction forming a glycosidic bond between the two monomers, glucose and fructose, okay? Now, in the background, what we have here is the fact that when plants are photosynthesizing, you know the equation, carbon dioxide plus water with the use of energy gives you glucose plus oxygen. Now, fructose is also another intermediate as part of that reaction. And we could go into the details, but essentially, you don't want to have lots of glucose floating around in the cell because it might just immediately get used up in respiration. Okay? So once you form glucose in photosynthesis from carbon dioxide and water, you don't then want to have lots of glucose to directly go into respiration and be used up again. Okay? So the formation of sucrose is one way to stop that from happening, and I guess what I really wanted to get across was why do we bother with sucrose anyway? Why is sucrose formed? Essentially, it's a way to prevent any formed glucose from being respired straight away. Okay, so the carbon dioxide and water by photosynthesis produces glucose. Fructose is also formed as part of that process, and therefore the two combine to form sucrose. And sucrose is what gets transported. So this is a diagram of the sucrose molecule. It's sucrose that gets transported around the plant so that other cells in the plant will also have access to sucrose in the case that they're not producing enough of it themselves. Okay? So what we are talking about here is if you have a plant, okay, so you've got plant there, stem, leaves, but also roots. Now, it's only the green parts of the plant that have chlorophyll and can photosynthesize, and the parts of the plant that are below the ground, they can't photosynthesize, but they're still made of cells, and those cells still need to have a source of glucose to be able to carry out all their metabolic processes via, obviously, getting the energy through respiration. Okay, so all the plant tissues need glucose or a source of glucose, but not all of the parts of the plant are photosynthesizing. So we need to have some way of transporting this molecule sucrose, which is produced via photosynthesis, and that process of movement of sucrose and other dissolved substances, in the watery part of the plant is called translocation. So it's this process that we'll be discussing. How does it work? We've just discussed the significance of it, but we will come back to that. Okay? Essentially, I just wanted to make the link between glucose and sucrose. We all are comfortable with the fact that glucose is the product of photosynthesis. Where does sucrose come from? I hope I've just explained that. Combined with fructose into sucrose so that it's not immediately, as soon as it formed, it's not used up in respiration, right? By converting it into this form, you stop that from happening, sort of. All right, guys? I'll come back and we will then discuss how translocation works. Okay, so let's just have a quick look at what we've discussed so far. We've discussed the fact that there are parts of the plant that are going to be producing lots of sucrose, and there'll be other parts of the plant that will not be producing lots of glucose. Okay? Now, how does stuff get from there to there? How does sucrose get from there to there? Because remember, these are also ATP requiring, or made of ATP requiring cells. They need to respire to obtain energy to drive their metabolic processes, protein synthesis, cell division, etc. All energy requiring processes. So how do they get that ATP if they're not producing their own sucrose? They get that sucrose from somewhere else, and then they respire that to form ATP. Okay, so how does it get from there to there? How does the sucrose move from there to there? It moves through tissue called phloem. Okay, now you've already gone through this structure of phloem. I'm just going to quickly give you the main details that are relevant to us right now. Okay, so what we'll be doing essentially, that was an ice cream van. What we'll be doing essentially is zooming in on a particular small part of this. Okay, and having a look. So, you know, in the stem there'll be many, you know, there'll be quite a lot of phloem tissue, and we're just going to be looking at one very tiny part of that. Okay, so the phloem tissue is made of two types of cells. The first are called sieve tube elements. Okay, so they're essentially cells joined end to end with each other, but the boundaries between the cells are broken so that essentially they allow continuous flow of material through them. Okay, all right, so very quickly then, sieve tube element, and these plates with holes in them are called sieve plates for obvious reasons. Okay, now these cells actually don't, as you can see, they're very simple. They don't have a nucleus, they don't seem to have any organelles either. So how do they survive? They survive because they have something called companion cells right next to them. Now these companion cells are essential for the survival of the cells of the sieve tube element. Okay, because what they have is a nucleus, and they also have organelles, and you know, in particular, relevance to us is the fact that they have many mitochondria. Right? And the other interesting thing is that their cell walls are broken so that things can flow from one cell into the other one. So things can flow from the companion cell into the sieve tube, right? They don't have to diffuse through the cell wall. Yep, these are gaps in the cell wall that allow continuous flow of things from the cytoplasm of the companion cell to the cytoplasm of the sieve tube elements. Okay, now this is really important because what that means is the sieve tube elements can actually act as a tube. You know, not you know, there's no organelles in here that will provide any kind of resistance to flow, any obstructions, and the companion cell outside of the sieve tube will carry out all the metabolic processes for the sieve tube elements. So the sieve tube elements can remain alive because all their kind of metabolic processes and proteins and things are coming from the companion cell. Okay, and the exchange of materials between these two cell types are made easy because of these gaps, and these gaps we forgot to name. are called plasmodesma. Okay? Many gaps are called plasmodesma. A singular is plasmodesmata. Okay, guys? So that's the kind of, this is the structure which is going to allow sucrose to move from a source to a sink. Okay? But how is it working? We'll have to zoom in a little bit to kind of look at the details. So I will rejoin you when I have redrawn my diagram. Okay? Right, guys, I'm back. I've drawn my diagram a little bit bigger and we're ready to go. I have had to move to a physics room, so even though I can feel the life draining out of me, I'm going to try and power through anyway. Okay, here we go. So, here we have things you should be able to recognize. We have our sieve tube elements, we have our companion cell, and remember, our phloem was, you know, our means by which sucrose could go from a place where it was being produced to a place where it was needed. Okay, so that's very simplistic terms. Okay, so sucrose, wherever it's being produced more than it's being used up, that's what we call a source, and wherever it's being used up more than it's produced, that's called a sink. I hope I got that right. Okay, now for simplicity's sake, what we'll say is that our source is somewhere like a leaf where there's lots of photosynthesis going on, and our sink is somewhere like the roots. Obviously, it's not always that simple. You know, there could be many parts of a plant which are not actively photosynthesizing a lot compared to the leaves. So, you know, they could equally act as sinks, and we'll look at different situations later as well. Okay, but for simplicity's sake, we're looking at how the phloem allows sucrose to go from somewhere where it's being produced to somewhere where it's being used up in general. Okay, so I think I'll need a little bit of space, so I'll get rid of this. Right. Okay, so the phloem is then our way that sucrose can move, and we're going to see how it's going to do that. Remember our sieve tube elements, remember our sieve plates with, you know, that allow the continuous flow of, you know, water essentially with lots of sucrose dissolved in it. You can refer to that as sap. And next to that, and remember the sieve tube element didn't have any organelles in it, and next to that we've got our companion cell with lots of mitochondria, big nucleus, fulfilling, you know, all the requirements of both the companion cell and the sieve tube element. And very importantly, the main thing that allows materials to flow through these two cells is the fact that they have gaps in their cell walls which allow movement of materials. Okay? So, and remember that they were called plasmodesma. Anyway, what's going to happen now is this. Now, remember I said that it was very important that these companion cells had lots of mitochondria. Why? I'll tell you why. Because mitochondria is how ATP is produced. And the way that sucrose is moved into the phloem tubes, into these tubes, is by active transport, essentially. All right? And or not active transport, but it's an active process that begins the sequence of events. So ADP is converted to ATP in the mitochondria during respiration. Now that ATP is used by a transport protein in the membrane of the companion cell. Okay, so what it does is it uses the energy of converting ATP back to ADP, and that releases energy, and that energy is used to pump protons, H+ ions, from in the cell to out of the cell. Okay, that's step one. And that's why we need lots of mitochondria, because the more mitochondria we have, the more ATP we can produce. The more ATP we can produce, the more we can break down back to ADP and use the energy released to pump out protons. And what that does is, over a certain amount of time, it leads to an accumulation of protons outside of the companion cell. Right? And what we have made, what we are creating using the energy from ATP, so it's an active process by which we are creating a diffusion gradient. All right? So lots of protons outside, few protons inside, and what that means is we have created a gradient of protons. Okay, it's a proton gradient. Now what you also have is another transport protein. Let's draw that different shape. Remember, these are all proteins. And what this protein does is it transports, actually what I should have done is draw lots of sucrose here. Remember, because we are looking at what's happening at a source, there's going to be lots of sucrose around here, okay? So, with lots of sucrose, lots of protons, because of, the sucrose has obviously come from other cells that are actively photosynthesizing. The protons have come from the companion cell. But what this protein does is a cotransporter, which means it needs to transport two things once at the same time. One of them on their own won't work. It needs the two together. So what happens is that a sucrose molecule is transported in with a proton. Okay? So I hope you can see that the more protons we pump out, the more sucrose can come in via the cotransporter. Because remember, the sucrose needs to be transported with a proton. Okay? So the two of them come in, protons come back in, obviously, but the sucrose is now in the cell. And if this process carries on, we're going to get a lot of sucrose in here. Okay, and that is going to create another diffusion gradient, because there's going to be lots of sucrose in here, and it will then diffuse into the Phloem tube. Okay, it will then diffuse into the Phloem tube through the plasmodesma. Okay, so now I hope you can see the important role of those gaps in the cell wall. Okay, diffusion of sucrose into the Phloem tube. Okay, so eventually we'll have lots of sucrose in here. All right, and because we've got lots of sucrose in here, it's going to lower the water potential in here, and then water is going to move in by osmosis. Okay. So it might help actually to kind of summarize the steps here. One, active transport used to pump out H+ from companion cells. That was the first point. And you know, I'm going to leave out a bit of details in the summary, but remember that's why it's important that these cells have lots of mitochondria. The energy comes from the hydrolysis of ATP to ADP, all right? Active transport, pump out the protons from companion cells. Okay, you generate a proton gradient, and 3, the cotransporter allows movement of H+ and sucrose down their concentration gradients or down diffusion gradients. Okay, so the cotransporter allows them both in there. 4, sucrose diffuses into sieve tube elements via plasmodesma. Right? Let me just check from where we can go. Yeah, we're okay. Okay, so, right. Now, I hope you followed me so far. A few more steps to go. As we've said, this lowers the water potential, so I hope this is now not going to go off screen. Check. Right. Okay, so the sucrose moving into the Phloem tube lowers the water potential in here, and so 6, water moves from the surrounding tissues into the Phloem tube. Okay, so water moves by osmosis from the surrounding tissues into the Phloem tube. Okay, lowers water potential. Water by osmosis moves from tissues into Phloem tube. Okay? Now, because of the movement of all this water in to the Phloem tube, the hydrostatic pressure increases, okay, and that is what causes the flow or the movement of all this material towards the sink. Okay, so 8 would be causes, actually let's just call that causes translocation. Is that going to go off the screen? Causes translocation towards the sink. All right, so let's just make a completely ridiculous situation that, say, here is the source and here is the sink. If the source is here, there's going to be high hydrostatic pressure because there's so much sucrose here, because the sucrose was high there at the source. If we have a sink down here, there's not going to be lots of sucrose down here. The hydrostatic pressure is going to be low, and therefore, in general, there's going to be a movement of all this material in this direction. Okay? Thus, we have movement of sucrose from a region where sucrose is being produced in general to other places where it's being not so much produced. Not great language there, okay? But I hope you get the idea. Okay, now let's just do a quick recap. And maybe it might help to be numbering the stages, yeah? So, first of all, we had the movement. Do I have a different color? First of all, we had the movement of protons out of the companion cell by active transport, as shown by this use of ATP. So that needs energy, therefore we have the mitochondria. That creates a proton gradient. The next thing was, we've got lots of sucrose out there, but sucrose, can't just diffuse through membranes. So we need a transport protein to move it in. But the transport protein that we have will only do it if a proton comes along with it. So the more protons we can pump out, the more sucrose we can bring in. So the cotransporter brings in H plus and sucrose together. Oh, I forgot to put the H plus over there. Okay, and therefore sucrose is now entering the cell. Now, if this happens many times, we're going to get a build-up, we're going to get a high concentration of sucrose in here, such that it will then diffuse into the sieve tube element. Okay? And when that happens, we are going to lower the water potential in this area, drawing in water by osmosis from the surrounding tissues. When we have a high hydrostatic pressure as a result of the movement of all that water, then it's going to start creating a movement, a translocation of all the solutes in that substance. Okay? And that's what we've talked about there. Alright guys? So, I hope that's made sense. If it's not clear, leave a comment or a question, below, and I can try and help you out, or maybe even redo the video so that it makes more sense. Yeah, but the exams are coming closer. We really got to get our head down and start working, so I hope this has been useful. What I'll do is now come back and we'll just have a quick discussion about sinks and sources, because it's not as simple as we've hinted at so far. Alright guys, see you in a bit. Sorry ladies and gents, forgot something important. What actually happens to the sucrose when it does get to a sink tissue? Maybe roots, another part of the plant that needs sucrose is not producing it. What happens when it actually gets there? So, hypothetical situation, let's say it's just got there. What happens next? It's in an area where there's not lots of sucrose around. What happens? Two things. It can move by diffusion out into the tissues, and then transport proteins on other cells will then be taking it up. So one is diffusion, but it can also leave the phloem tubes by active transport. Okay, so this is what happens when it gets to the sink tissues. We discussed what happens at the source. That's how sucrose moves into the phloem tubes and how the movement is generated. But when it gets to the sink tissues, that's when it either diffuses out of the phloem tissues because it's no longer in an area of high sucrose levels. It's in an area where there's not a lot of sucrose around, where sucrose is being used up. So it might diffuse out. It might also move out by active transport. All right? Okay, and I think that really is it this time. Thank you. Just a quick kind of final few points. It's about sinks and sources. Okay? Now we've kind of simplified it by saying that, in general, leaves would be sources, and things like roots would be sinks. Yeah, and in general, that's true. Right, because these guys are carrying out photosynthesis, producing glucose, converting into sucrose, and having lots of sucrose, and therefore it's a source. Okay, now when there's lots of sunlight and you have leaves which are actively photosynthesizing, that's all true. Okay, but remember that these roots, when they get that sucrose, a lot of that would be stored. Yeah. So sucrose, when it gets to sinks such as roots, it can be stored. Sucrose can be stored, for example, as starch. Why would it do that? Because remember, at some point, you know, in the year, you know, a lot of plants, deciduous plants, will lose their leaves, okay, and they don't really have any active photosynthesis going on up there, right? So, but that doesn't mean that the plant is dead. It still has living cells, but no photosynthesis going on. So, where is the sucrose going to come from? It will come from all that stored starch in the roots. All that stored starch will now be broken down back to glucose, that glucose converted back to sucrose, and now it might need to go in the opposite direction because, you know, we don't have any photosynthesis going on. So now, when we don't have, well, you know, the sun still comes out, but the temperatures are so low, leaves are not there, no photosynthesis going on. At different times of the year then, the roles might reverse. You might get roots now being the source. Yeah? The roots are now being the source, starch is being broken down in the roots, all that sucrose that the roots stored up from before when they were the sink, used, you know, absorbed it, converted it into starch for storage. Now it's being released back because there's no active photosynthesis going on. We need sucrose still for the living cells. So now the starch is being broken down back to glucose, converted to sucrose, and now there's a lot of sucrose down here, but not a lot up here. So now this is the source, and this is now the sink. Okay, so just bear that in mind. And even in a regular plant with its leaves, you know, I'm sure it's not likely to be as simple as roots are sinks, leaves are sources. That is true, but you know, even, you know, one part of the plant may be having lots of intense sunlight, whereas another part of the plant might be in the shade. In that case, maybe it might be the case that this leaf is also a sink compared to that being a source. It all depends on which part of the plant is able to generate more sucrose. Okay, so while, you know, in most situations leaves are sources and roots are sinks, remember that, you know, it's not always that simple. Yeah. All right, guys. Good luck. Remember, any questions? Now is the time. Few weeks left. Get your revision done, and good luck.